Thank you. Very happy to be here. So this talk will focus on something quite different than what you've heard thus far. The data I'm going to talk about focus on parents who are at increased genetic liability to autism or Fragile X syndrome. And in the case of parents of individuals with Fragile X, they will be carriers of FMR1 in its premutation state. And FMR1 is the gene that causes Fragile X. I'll give you some background on that in a moment. I guess just going into this and sort of thinking about links with the other talks, an underlying motivation for this cross-population family study type approach is really to identify more homogeneous subgroups, which are always helpful for diagnostic and etiologic research. Okay, so there's a lot of overlap between autism and Fragile X. Fragile X syndrome is the most common inherited form of intellectual disability and the most common known genetic cause of autism. Prevalence rates range, conservative prevalence rates range from anywhere between 25 and 50% of individuals with Fragile X also receiving a full diagnosis of autism. So this suggests that mutations in the FMR1 gene, which causes Fragile X, may be a highly important risk for autism. And so while efforts to uncover the etiologic mechanisms in autism are quite often confounded by multiple unknown etiologies, genetically defined syndromes like Fragile X can provide this really uncommon and useful opportunity to examine gene-brain behavior associations in a more etiologically homogeneous condition. So studying genetically defined conditions such as Fragile X that show overlap with autism can really be used as a foothold for identifying, again, these more homogeneous subgroups for studies on treatment, for studies on etiology, brain behavior relationships, so on and so forth. Okay, so we're also taking a broader phenotype approach focusing on parents who are genetic carriers. This is another approach for really trying to refine the complexity of the phenotype, so distilling down to genetically meaningful features that may aggregate among family members who are carriers, but are much more subtle in expression, not typically associated with any functional impairments, but qualitatively similar to what you see in these disabilities. So in the autism literature, there have been a number of family genetic studies that have shown repeatedly that there's a constellation of clinical and language features that mirror in quality what we see in autism, but are much more subtle in expression. And so we were interested in studying these features among people with parents of individuals with autism, in contrast to parents of individuals with Fragile X who are carriers of this autism candidate gene, to see if there was any overlap. And then this pedigree here is just to illustrate how useful it can be taking this family study approach where you're studying these clinical phenotypes in relatives. So knowing, for instance, that you see subtle language features in one parent, but not subtle social deficits, could suggest that these features disaggregate independently in families and could relate to separate genetic effects. Okay, straight to the participants. We studied moms, and this is because we know a lot about the inheritance patterns in Fragile X, and all moms of a person with Fragile X are carriers of the Fragile X gene, FMR1, in its pre-mutation state, unless, of course, they are carriers of the full mutation, but we screened for that. So we had this well-defined genetically defined population of mothers of children with Fragile X. We included, and so we wanted to match on gender, so we included only mothers of individuals with idiopathic autism and mothers of typically developing children who were screened for any sort of developmental difficulties that could be indicative of autism. And the mother groups were matched on age, socioeconomic status, and race and ethnicity. Okay, so we were looking at, I'll get to the assessments in a moment, but our question was, is there, in comparing these groups along these different clinical measures, are we going to see any evidence for expression of the subtle autism features that have been documented in prior studies of autism among these FMR1 carriers? So we hypothesized that if FMR1 is playing a role in autism symptomatology, that yes, we would see that. And so we would see elevated rates of these features in both the autism and Fragile X mother groups relative to controls. So very briefly, these assessments have been used repeatedly in family study research, and I'm happy to give you more details afterwards. But for personality, we use the Modified Personality Assessment Schedule. This is an in-depth interview where you solicit concrete behavioral examples of these different traits that that have been described as constituting a broad autism phenotype. So socially aloof personality, some difficulty with flexibility, perfectionistic traits, and then we wanted to elicit a language sample to measure pragmatic language violations, which is also a key feature of this broad autism phenotype. So we used Rebecca Landa's pragmatic rating scale to rate pragmatic language use in a naturalistic conversational sample. And so for both of these, interviews and assessments were videotaped and then rated by raters who were blind to group status, and determined the number of individuals rating positive or negative for each of the personality features, and then for language, the rates of pragmatic language violations. And then finally, we were really curious to see if number one, if we see elevated rates of these features, would they be correlated at all with what we see in kids? So might kids with moms who are showing features of the BAP, the broad autism phenotype, also should be more likely to show features of autism? And so we administered John Constantino's social responsiveness scale to assess autism symptomatology in children. Okay, very quickly. Results. Okay, so on the left here are the personality features. The red bars are the Fragile X parent group, blue is autism parent group, and yellow are controls. For each of these personality features, overly conscientious, inflexibility, and this aloof social disposition, we had significantly more parents in the Fragile X and autism groups rating as positive than we did in controls, and there were actually no controls who rated as inflexible in this group. Okay, similar pattern for pragmatic language violations, and I should say pragmatic language violations are things like having difficulty with conversation, so going on tangents, going on too long, not being able to correct a message if your interlocutor hasn't understood it. So this is the rate of the mean number of pragmatic language violations committed by each group, and you can see both the Fragile X and autism groups are committing many more pragmatic language violations in conversation. Okay, and then finally, these are our correlations here. So 3 out of the 4 parent characteristics that I just described for you correlated significantly with autism symptomatology in the children. So I'm just going to jump to the conclusions for this. I think in general, these results showing significant phenotypic overlap between these two groups provide further evidence for not only the overlap of autism and Fragile X, but also for the role of FMR1 in subtle autism symptomatology among parents. So if you look down here, this is just a schematic of the different mutation categories for Fragile X. So full mutation, this would be an individual with Fragile X. These are the moms we were looking at. So they had a pre-mutated state. And there have been a number of phenotypes associated with this. Premature ovarian insufficiency, anxiety, depression. Now it looks like autism among young males with the premutation. Autism seems more common, and this study would suggest that among mothers with the premutation, the broad autism phenotype features are also more common. So it's exciting about the potential implications of this candidate gene in the features associated with autism, and I think. Together, this work can really help us provide clues to autism genetics and ultimately identifying genetically meaningful subgroups, which could also help for treatment formulation. Thank you.